Welcome to Ask and Learn.
Hello students, in this question we are being asked various questions regarding the DNA fingerprinting of a person who has been in prison for 10 years. The first one is to identify and analyze DNA from a person who has been in prison for 10 years. For that, the following steps would be taken. First one will be collection of physical evidence. And this will be the first step to collect any physical evidence that contains the individual's DNA, such as blood, hair, or saliva sample. Then second would be extraction of DNA. And then DNA can be extracted from the sample which is collected, physically collected from the scene of a crime by chemical and physical method. And then after that, we have to quantify the DNA. So after the extraction of the DNA, extraction of the DNA, quantity is or the amount of DNA is measured to ensure that there is enough for further analysis. And after that, amplification of DNA. The extracted DNA is then amplified using a technique called as polymerase chain reaction, which creates multiple copies of the specific regions of the DNA. And after that, separation and detection of DNA fragment. And these separation of DNA fragment is done by a gel electrophoresis or a capillary electrophoresis, and then detecting using a fluorescent dye. Then after that, we have to comparison of DNA profile. The DNA profile obtained from the evidence is compared to the DNA profile of the individual in question to determine if there is a match or not. Second question is regarding the process of identifying DNA forensic lab. It involves following steps. It is DNA extraction, the DNA is extracted from the physical evidence using chemical and physical methods, and other steps which are involved in the process of identifying and analyzing the DNA. The same for this step also, which includes DNA quantification, DNA amplification, DNA separation and detection, then DNA profile generation. So it is the same as previously discussed for the process of identifying DNA in a forensic lab. Another question also is to consider that the evidence is over 10 years old. Once the DNA evidence has been identified, what is the process of analyzing DNA? What challenges exist that can make your analysis inadmissible? Oh, explain. Considering that the evidence is over 10 years old, there may be many challenges in obtaining sufficient high or sufficient amount of high quality DNA for analysis purposes, and degradation and contamination of DNA sample may occur over time, which can affect the accuracy and reliability of the result. Another question asked is explain how or tissue matches A and explain why it is important to utilize different DNA testing methods. The process of analyzing DNA involves comparing the DNA profile obtained from the evidence and the DNA profile of the individual in question. If there is a match, it may suggest that the person was a crime or incident, and the challenge that can make the analysis inadmissible in a court include, as already mentioned, that contamination of the DNA sample or and degradation of the DNA sample over time. And it is important to utilize different DNA testing methods because some methods may be more suitable for specific types of samples or situations, and different methods may also provide different levels of sensitivity, accuracy, reliability, and some methods may be preferred over others due to factors such as cost, time, and type of sample being analyzed. Another question is regarding the pattern testing. Paternity testing can also be done using the DNA sample, which the DNA sample from a child in question is taken and it is compared with father, suspected father, because we take the sample of the father as well and compare the child's DNA profile with father profile. And if it matches, then that means father, the man in question is the father of the child. I hope you found the video useful. Thank you.